All right. Hello, everyone, and welcome to today's keynote webinar titled High Yield Vesicle Package Recombinant Protein Production from E. coli. This webinar is a part of the 5th annual event in the Bioprocessing Virtual Event Series. I am Sydney McNeil of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We do encourage you to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of the navigation bar or from the lobby of the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. I now present today's speaker, Dan Mulvihill, PhD, Professor, Cell and Molecular Biology, University of Kent. For a complete biography on our speakers, please visit the Presenter tab from the menu at the left of your screen. Dr. Mulvihill, you may now begin your presentation.
Okay, thank you very much, and welcome everybody. So today I'm going to talk to you today about some research, recent research that we've just published a month or so ago now, where we describe a brand new system for generating protein from vesicles for the bacteria E. coli. So high yield vesicle packaged recombinant protein production from E. coli. So recombinant proteins are incredibly important. They're incredibly important for biological research. They're also incredibly important for many different industries. So health industries, agri-science, energy, you name it, there are proteins involved in it. So the global annual recombinant protein market is vast. Almost $2 billion a couple of years ago, last year. And it's expected to grow continually. So, and for many of the proteins that are produced, this simple bacteria, E. coli, so it's a gram-negative bacteria. It's the organism of choice, and so it actually approximately one third of all recombinant proteins, industrially produced recombinant proteins, are generated in E. coli. So the reason this is because E. coli, it's very easy to grow. It's very quick to grow. So it when it's actively growing, it doubles. It grows and divides once every about 20 or so minutes. So it's very quick to grow. It's very cheap to grow because the media you can grow, you can just make it in the lab. You don't have to buy it in, and you don't need such detailed and complicated sterile techniques that you do when growing tissue culture. However, there are a number of challenges for growing recombinant proteins in bacteria. So there are some proteins that you cannot produce. So there are some more complex human proteins that are very challenging, so you just can't produce them in bacteria. But also, if you just kind of look at these images that I present here, they highlight some of one of the beautiful, on the left-hand side is one of the beautiful pieces of art by David Goodsell that highlights just what is inside the cell wall. And you've got multiple layers of membrane, as well as this layer called a peptidoglycan layer, which is a mix of amino acids and sugars. And you've got to smash all of this up in order to get into that protein goodness, which is inside the cell. So on the right-hand side here, I think we've got an image. This is an E. coli. This is a super resolution image that we took from within the laboratory, where in green, you've got the inner membrane labeled, and on the right-hand side, and in magenta or pink, you can see the outer membrane. So you've got to smash through those, you've got to burst those, and then you've got to get through that peptidoglycan layer, crack it all open in order for you to get that protein out. So a lot of research over the last decade or more has been spent on looking at ways to secrete proteins from E. coli. So get the protein to export them from the bacterial cell. And there are a number of known transporters or mutants that have been hijacked, some of the wild type transporting mechanisms from the bacterial cell that have been hijacked by biotech labs. So the TAT system, the SEC system for secreting for different proteins, and they work to some extent for some proteins. However, but what we've done is, and they work for very specific proteins in general, but we've developed a system a very generic system that you can apply to all sorts of proteins, lots of different types of proteins. And what happens is it packages these proteins in little membrane-bound vesicles that then get exported out into the culture media. So you don't have to smash all the cell wall up. So they just get delivered. So these vesicles, they're really nice because they also provide a really stable environment for the proteins as well. Okay, so they're because they're all inside it, they've got all the good, all the right pH, all the right kind of osmotic potential as you'd find within a bacterial cell. So the proteins are kept in a native state as possible, so you can store them for a long time. And so as the vesicles are being exported from the cell into the media, it makes purification really easy. So all you've got to do is just get rid of the cells, spin the cells down, and in the media, you've got the vesicles just sitting there. So we came across this, like many of the many of the significant breakthroughs in science over the decades, over the centuries it's been through luck. There's an awful lot of luck involved in this, but I guess it's identifying when you get those lucky breaks is the important thing. But also this has been it's a side project of a side project of a side project from one of the main interests within the lab. But anyway, so I'm not going to go into how we got onto this project, but it's sometimes, as my old PhD supervisor always used to say, you need to stay focused. It's always good to have something on the back burner, but it's just getting that balance right. And this is a really nice example or demonstration where working on those side projects can actually lead to some good things. So the idea was, on the number of the slides that I'm going to show you today, you might often see this VNP. So that stands for vesicle nucleating peptide, which I'll describe more as the project progresses. So the way this project works is we identified a sequence from an endogenous mammalian protein. It's a neuronal protein, and there's a region of it. So that is enough to drive vesicle production in bacteria. And we just came across this during we were developing a drug screen at the time, and it was a very serendipitous discovery that we just happened to look down the microscope and thought, oh look, there are some nice looking vesicles. I wonder if we can do something with those. So then we got into I went to an industrial workshop conference, like a club that we have in the UK, between the research that funded by one of the research councils, and I got talking to couple of researchers from Fujifilm Diosynth Biotechnologies, and we talked about the project, and they were interested in this and actually applying this in their production system. So we sought funding, just a little bit of funding to get some proof of concept data to take this forward. And what we did was, so as we can see in this figure I can show you here, is we have our vesicle nucleating peptide in blue. And then next to that, we fused to it the DNA that encodes for one of the fluorescent proteins. It's called neon green. And then to the C-terminal of that, we fused a recombinant protein where it's a therapeutic, so human growth hormone, which is, it's of great interest. It's of great use to people. And actually, the problem is within bacteria, it can be quite challenging to express because it doesn't generally fold correctly. It's got things like disulfide bonds within the protein structure, which can be challenging to get to form correctly within a protein. Anyway, so this was the idea, and we thought, can we get our system? Can we get this proof of concept? Can we get it to work? And then from this, where we did show it worked. And then from this, we got further funding. And actually, we're just about to start the next level of funding beyond that now. So here's kind of a quick video And then on the right, you can see a fluorescent filled vesicle as it's see it grow, and then suddenly gets released into the media. It's very quick. Okay. But let's go on to here. So let's actually look at some of the images. So on the left-hand side here, so now we've got our vesicle nucleating peptide. And during our study over the last, I guess, 7 years now, we've been working on this. We've been able to narrow it down to a small vesicle nucleating peptide sequence. So it's down to, optimally, it's between 20 and 38 amino acids long. And what we happen, so we fused that to our protein of interest, and often is the case when we were developing it, it was a fluorescent protein, and we can look at the formation of these extracellular membrane-bound vesicles. The reason we know they're membrane-bound is, so if you look at figure A on the left here, this is where we've got fluorescently labeled outer membranes, and we can see the formation of these vesicles on the end and along the edge of the cell. Then, and the B, C, and D, these are negative stained EM images where we're looking at the surface of the bacterial cell. And in B, you can see somehow our peptide, our vesicle nucleating peptide, is causing this membrane to curve outwards. And eventually this will continue to curve outwards, and we'll see these vesicles that you can see in C. So the image in D, again, this is an electron microscope image where we've got a couple of bacteria. So those are these really electron dense regions you can see here in black. We let them grow on a grid, on an EM grid, and they were expressing the nucleating peptide, and you can see them just keep on pumping out these vesicle nucleated, so these vesicles overnight. And so this has got, we can see all these vesicles form. Okay. So that's what we've got, the vesicles. We can make the vesicles. What about our protein of interest? Can we get things into them? So if we then actually look now at our vesicle nucleating peptide, which is tagged to a fluorescent protein. In this case, it's called neon green. So it's very nice. It's a fluorescent protein of choice within the lab because it's monomeric and it's very bright and it's quite photostable too. So in green, so in neon green, we've got fused to our vesicle nucleating peptide, and we can see it here in E. We can see the green of the protein. It's in the cytosol of the bacterial cell, but we can see it concentrating at the cell surface, at the cell wall, but we can see it really concentrating in the lumen of this vesicle. So in magenta or pink here, we can see this is where the outer membrane has been labeled. So in F, this is where we've got an immuno-EM image, where we've got a cross-section through a bacterial cell which has got a vesicle, just like in E, but in the gold labeled, see these black dots here in F? These are these are antibodies. It shows you where the VNP labeled M neon green is, and you can see it's all within the lumen, or within the center of this vesicle here. So in G and 8, so G, this is where we've just got we've harvested and concentrated up some of the vesicles, and just here's an EM image to show what they look like. H, this is a fluorescence image to show that, yep, we've got lots and lots of green stuff inside our vesicles. So it all seems to be working nicely. So, and following on from this, so, okay, again, let's look at these cells in culture. So here's the same cells where we've got them in pre-induction. So before we've induced the expression of our vesicle nucleated peptide labeled neon green protein. In the middle, the bit on this post-induction image, this is where you can see it's really green because it's fluorescent protein. We're just shining blue light at it, so it makes it glow green. And this is the culture. You've got an overnight culture, and we've got all the cells there. And then in the right-hand image, supernatant. So what we've done here is we've just centrifuged the cells out of the culture. So all we've got here is the media which contains the vesicles. Okay, so the cells are out of it. We've just got vesicles. Very simple purification. We've got rid of the cells. On the right-hand side here, this is where we've got a Kamasi-stained SDS page gel image where we've run the whole culture on it. So this is the pre-induction, so that's pre-I. Post-I, that's the post-induction, so that correlates with these two images here, and that's the supernatant there, which correlates to the image on the right-hand side. And you can see, and this really stronger band there, which is just running over 30 kilodaltons in size, that correlates with the neon green protein. So it's pretty pure. It gets it out the cell. So already, I mean, for many applications, this is pure enough for you to be able to do what you want to do. Obviously, you know, if you want to go and go on to GMP or therapeutic studies with these proteins, you'll need to go further up purification, of course. But anyway, so it's quite easy to isolate them and purify them from the culture. So what we're doing now, this is our current method, and we're in the middle of developing a simplified method to purify these vesicles, but concentrate them as well. So this is basically our pipeline. So we culture ourselves, we do a transformation of bacteria with our construct, which has our VNP labeled protein of interest, a gene encoding for our VNP labeled protein of interest. The following morning, we set up a starter culture. And then we set up a main culture overnight. And this culture has to be grown in very optimal conditions, very specific. So it needs good oxygenation. So you need good volume to surface area ratio when you're growing them in flasks like this. So you need good high oxygenation. You need good temperature. So for most of the proteins we express, we get optimal vesicle expression at 37 degrees. Although I'll talk about later on, we've got a couple of variants now where we can go down to lower temperatures, and we do not induce the full strength expression from a standard promoter using E. coli. So for example, if you were to use the T7 promoter using IPTG to induce, we don't use full induction using 100 micrograms per mil, which is standard practice. We actually use something like between 10 and 20 micrograms per mil IPTG to induce. So then we've got our culture, overnight culture. We spin down the cells. Then we filter the supernatant, and we can see the cells stuck on the filter. They'll purify the vesicles, onto this filter, and then we can do whatever we want with them. They're all concentrated up. So this is what they look like. So overnight culture, here's the cells, all nice and green, because in this case, they're expressing neon green labeled VMP. Here's the one that we've got rid of the cells. We can see just vesicles. And here's when we've concentrated them on the right-hand side. We can see really concentrated vesicles. Nice and easy. Okay, and finally, if you then just to confirm the purity of these samples. So if we were to take some of the culture here without spinning out without spinning out the cells. So if we were to plate them out onto a plate that has the antibiotic resistance, we get nice colonies, because they've still got the plasmid in them. It confirms it. If we were to put some of the media, the centrifuge media onto a plate which lacks antibiotics, so there's no selection whatsoever, we get no colonies at all. And that's what we see. So it shows it's pure enough. We've got rid of the bacteria anyway. And on the right-hand side, this is just the fluorescent image of the purified vesicles where we shine blue light at it to show that they're fluorescing. And here, purified vesicles. We like to do some EM images because, I'm a microscopist, I'm a cell biologist, so I like to look at things down scopes anyway. But actually to confirm that these vesicles, they have structural integrity, they look right, they look like we'd expect them to look. And they're quite heterogeneous in size. So, they go anything from, I'd say, about 200 nanometers in diameter to about a micron or so. And some are a little larger, but that's the standard size of them. So the vesicles are really quite stable. And this is really nice because, and this is really good for storage. But also, and we wanted to make sure, as our image I showed you earlier, it suggested that all of our recombinant protein is on the inside of the vesicle. It's not pointing out of the vesicle. So to confirm this, we just did a bursting experiment. So we purified the vesicle and we undertook them we subjected them to immuno-EM labeling, just like we did before, using an antibody against the neon green protein. And on the left-hand side, you can see all the vesicles, but there's no gold labeling as we would expect. However, first, so we subject them to an osmotic shock. You can see there's immunogold labeling everywhere. So it's releasing the protein. So it confirms that all that protein is inside those vesicles, or inside the lumen. And they're pretty stable. So we've got the protein, we've got the vesicles, and and then this allows us to do, it simplifies the downstream processing. So here's just an example of experiment where we've got VMP labeled proteins. We've purified, we've got from vesicles, we've taken the vesicles, we've concentrated them, and we've run them on a gel. And this is the concentration that we're getting. This is what the band looks like. This is our protein of interest that we're purifying. And these are contaminating bands. And when we do proteomics of these experiments, we do tryptic digest of the contents and do proteomics of them, we can always determine that there's between 40 and 60% of the total protein within these vesicles are the protein of interest, are the protein that we've labeled with the vesicle nucleating peptide. Great. One thing that's always, if you've got a label like this and you've got something that's interacting with the membrane, does that affect the does that affect the viability of the bacteria? So if you're expressing something that's interacting with the membrane and forming vesicles, an obvious question is, are we making the cells burst? So with the concentration of IPTG that we're using, this is why it's really important, the IPTG levels, that we find that we do not get any measurable toxicity to the bacterial cells. Okay, so if you go higher, obviously you will, because then I think what happens there, you start making too many vesicles and eventually the cells do burst. But if you go at the concentrations that we use, you see there is no negative impact. There isn't any negative impact. Let me just correct that double negative there. There isn't a negative impact upon the growth of the cells, the viability of the cells. And actually, if anything, it increases the viability of the cells, as you can see in these graphs here. So here we've got a technology, we've got a proof of concept, and that kind of works nicely with neon green. But what about proteins of interest? What about industrially relevant proteins and therapeutic molecules that obviously people like Fujifilm and many other companies could be interested in? So we've tested something like 70 different proteins, maybe even actually somewhere between 70 and 100 now. I can't remember the exact number. Oh, there are a lot of proteins we looked at. So we've tested a range of them, and to see if there was impact on the yield, make sure they were exported, and make sure they were soluble. Because if we can getting an insoluble protein, okay, you can do things with that. You can resolubilize it, but actually, life is a lot simpler if you can just make a soluble protein. So we did that here, and here's just a few of the examples. So we've got three proteins here which you can see on the gel, on the Kamasi stain gel on that K. So we've got DARP. So that's an anchorin repeat protein. We've got uricase, so it's a therapeutic, it's involved in the breakdown of uric acid. And we've got stephin A, which is a protease. So differences in sizes, differences in shape, structure, and activity. And all of those, so in each of these lanes, so it goes pre-induction, post-induction, and then within the vesicles, the F, okay? That's the filtered media. And you can see for all of them, there's in each of these examples, just showing that there. And then actually, if we look at the yield of soluble protein, and this was a bit of a surprise for us because we weren't expecting any real significant change in yield. We just thought, our end game, our aim was to, just export the protein to make life simple. But actually, there's a dramatic increase in yield. In some cases, over 100-fold increase in yield. So if we just look at the example here, DARP, so neon green, for example, here, you can see there's a big increase. DARPin, so this is DARPin off 7, I think, that binds to MBP. So that, if you just express, so this is all these experiments we're undertaking in shaking flask cultures. If you just have the DARP by itself in a bacteria, express it, standard techniques, you'll get something like 50 milligrams of DARP per liter of culture media, okay, using optimal conditions. Using the same conditions with our VNP labeled DARP, we get close to a gram of protein per liter, okay? So this is the kind of concentrations that you hope to get when you're using fermenters or bioreactors. But we're getting this routinely with shaking flask cultures, and you can see that with all of these proteins here, similar kind of increases. So yeah, so on these images, on these histograms I'm showing you here, the shaded color is the exported region, exported protein, and in the light region, so the unshaded region, that's the protein that remains within the cytosol. So it changes according to some of them. And these green images we've got here, these are also got neon green labels on them, but there's some interesting ones here right at the bottom, which I'll come back to later. There's etanercept. So this is an IgG fusion, requires disulfide bonds. And for this one here, we are, we're getting formation, we're getting lovely soluble protein, but the majority of it is within the cytosol. Similarly, with that human growth hormone that we described in our first proof of concept experiment, we can see we're getting huge yield of this protein. But this is a standard BL21 strain, BL21 DE3 E. coli strain. But a proportion of it is being exported, but the majority of it is internal. So we'll come back to that later. So our proteins, they are exported, and we're getting soluble protein, and we want to just determine to make sure that that tag that we have, it wasn't keeping the protein soluble. Okay, we want to make sure that was the case. So we cleaved it off. So we introduced a TEV cleavage site, so we can cut it off, and our protein all remains soluble within the supernatant. Okay, so it remains soluble upon cleavage. So the VMP, it enhances solubility, it's enhancing its expression, it's allowing it to be exported, but once it's formed and it's soluble, it's not necessary or not required to keep it soluble. And also, all of these experiments I've shown you to date, they are up to here on this presentation. They have been undertaken in the lab using shaking flasks, but up in food, but within at Fujifilm, they're obviously in terms of industrial applications, we're interested and want to look at whether or not this works in bioreactors and within specific conditions that we've optimized now. You can get really good within fermenter, within bioreactor cultures. Okay, and this is just one example where I think in this case here, I can't actually remember what we're looking at here. I think it's DARP again in this case, in this example. Okay, so it works with bioreactors. So that's one thing. So it seems to work. But another interesting thing that we found is it allows production of toxic proteins. So human growth hormones, some of those examples I've shown you up there, cells don't like it, and they really reduces the growth. And we thought, okay, can we push this a little bit further? What's probably one of the most toxic proteins we can think of? Okay, what about DNAs? Because that's going to chop up the DNA within a bacterial cell, and that's going to, it's no longer going to remain viable then. So we did the experiment. We made a construct which is absolutely switched off in the absence of induction, or it switches on, so we can really control it. There's no leakiness with the promoter. We did it without the VMP and with the VMP. And on the graph on the left-hand side here, we can see the viability. There's no growth. We're looking at growth. So we're looking on the y-axis in this example. There's no growth at all. Whereas if we put the VMP on, so the filled circles, so we've got VMP DNAs, we're getting really good. The cells are growing as normal. And again, if we look on the right-hand side, we can look at the in the absence of VMP, you don't get any real measurable expression of the DNAs. However, with the VMP, you get really good measurable expression. And so this is an experiment I did in the lab, which I quite enjoyed. I don't get to do it very often. Is actually I purified that protein and showed that the DNA is digested by that DNA, but DNA's protein, so it's active. So we've undertaken this for a number of proteins that are generally challenging to bacteria, like capsid proteins. So normally it's insoluble or toxic to the bacteria. So we've used that, and we're actually using it in vaccine trials in fish. And also there are nanobodies, so camelid antibodies. So a number of those are insoluble, but we found with our tagging technology, it allows them to be soluble. And we can get pretty decent yields, like almost 200 milligrams per liter. So we make proteins. Is it active protein? So we know they're folded correctly. We know they're interacting correctly. We know they can be cleaved and they can bind to ligands. But what about enzymatic activity? So one of the proteins that we isolated and purified was uricase. So it's involved in breaking down uric acid. And so we did an experiment where we compared the enzymatic activity of uricase which is just purified from a cell pellet, as you normally would. So that's in red, and compared it with the activity with VNP uricase, which is isolated from vesicles. So in this experiment, we're looking at the breakdown of uric acid in the lab, and they have similar, if not equivalent, they have equivalent enzymatic activity. So we did the same experiment then, so that we did here, but this time we did it on vesicles that we'd stored for, it says, yeah, for 4 months in the fridge. Does they maintain their enzymatic activity? And this experiment here, we're actually using a stop flow kinetic, so we can look at it more rapid kinetics of the reaction, and actually it keeps its activity. So compared to purified protein, which is stored in the fridge for 4 months, that loses its activity over time. If you store it in our vesicles, just keep it in the vesicles, and then when you can do the experiment or do your analysis or do whatever you're going to do, crack open the vesicles. We use sonication, it's just easier. Crack open the protein and then purify the protein, and it's just as active as it was on day one. Okay. So enzymatically active also, so we've done it lots of different proteins. Here's just a table. Actually, this is quite obsolete now, this table, but we've lots of different proteins we looked at. We've also identified or shown that we can purify protein complexes using this technology. So here we've done a bimolecular fluorescence complementation, and what this is you've got a fluorescent protein, which is basically cut in half. And on one half, we have the DNA encoding for the amino terminus of the gene. We put one VMP onto it, so A. And then the DNA that encodes for the second half of that fluorescent protein, we put onto another VMP. So on B, I can see on this image here. And if they are together inside a vesicle, they'll fluoresce yellow. So if there is fluorescence within the vesicles or in the media, that illustrates or indicates, it's more than a strong indication that you've got a complex of these two molecules within the same vesicle, right? And we can see that, and we can actually help enhance that dimerization. We can introduce a leucine zipper as well, so a dimerization sequence. So we can get protein complexes, and this is also consistent with the work we did with etanercept. So etanercept is an IgG dimer. It's a fusion between TNF and an IgG. It forms a dimer up in the IgG region. It forms these disulfide bonds, so it forms a homodimer. And we'd be able to purify this. We know that it forms a dimer because we've done add DTT. We get a really tight band at the expected size, about 95 kilodaltons, which you can see there. And you've got this multimer in the absence of DTT. Then we can purify on protein A. So it's a functional protein antibody. And actually, again, just like we did with the previous experiments, I think with the DARP I showed you earlier, we can cleave off the whole neon green and the VMP sequence off of the amino-terminus, and the protein remains soluble and it binds to that protein A. Okay, so it's soluble. It contains the disulfide bonds when we have it there. We put it with the VMP. It's a functional IgG because it binds to the protein A, and it remains functional after cleavage of that VMP neon green tag. Great. But back to that original, those graphs I showed you, those histograms. With etanercept and human growth hormone, we saw that the protein was going inwards. And actually, when we do the EM images of the cells expressing etanercept, we see these vesicles, rather than going outwards, they form inward vesicles. And when we do the immuno-EM for these cells, we can see that these vesicles are filled with the neon green labeled etanercept. Okay, this is just looking at the yields from the culture. So that's okay, it's working nicely. Actually, we get similar kind of yield. So remind you, that's where you get internalization with the human growth hormone as well. And so internalization, we can get the same thing if we introduce, because sometimes it's actually quite nice to have dimers inside the bacteria. There are some applications that we're exploring currently that would be good to do. So we thought, can we induce dimers by introducing that VMP dimers by using a leucine zipper? To confirm that we could do that, we express some VMP dimers with the neon green on it, and compared them. So VMP leucine zipper M neon green fusion, or their and compare that with VMP neon green by itself. And yes, biochemically, we can see it forms a dimer. And actually, when we look at these cells down the microscope. So by the EM here, we can see in the middle, you can see that we've got the neon green labeled vesicles on the inside. And here on the right-hand side, we've got some super resolution images where we fluorescently labeled the inner membrane of the bacteria. And we can see we get these vesicles. And here is a time lapse. These vesicles are dynamic, and we see them moving around. The vesicles, they get released, and off they go. They do what they're meant to do. So how does it all work? So this is an ongoing bit of analysis and we've got onward going funding to explore this in more detail, and we're applying this to different model systems, I guess. But how does it work? So our helix, our VMP, is an alpha helix, and it's amphithalic helix. So that is, it stabilizes that alpha helical structure once it interacts with the membrane. You can see that through this circular dichroism experiment here. So if you have the VMP by itself, we don't have a peak here at about 210 nanometers wavelength. But then as soon as we add the lipid, we get a really nice peak there, that's consistent for alpha helical proteins. Then we know it binds to membranes, but here we've shown specifically using a number of biochemical assays. Again, so here's some fluorescence binding assays to different lipids. And on the right-hand side, we've got some fluorescence lifetime and imaging, some FLIM-FRET experiments, and we've shown that it specifically interacts with the inner membrane of the bacteria. It doesn't, the vesicle nucleating peptide does not interact with the outer membrane. Well, in our hands anyway, we don't detect it. We certainly see it interacting with the inner membrane. And now we're doing a whole series of lipidomics experiments to get more characterization about the state of the bacteria when they're producing the vesicles, and also the vesicles themselves, so we can get a better understanding about, does the lipid have to be in a certain state to get the best yield of vesicles and the best yield of proteins. So how does it how does it work? So this is our current model. It's a very naive model, but this is what we kind of think is going on. We've got our vesicle nucleating peptide in blue. It interacts with the membrane, the inner membrane of the bacteria. Once you've got a critical concentration of these peptides that are interacting here, so on this cartoon we've got neon green stuck on it. So it's nice beta barrel structure you can see there. Anyway, so once you've got a critical concentration of the vesicle nucleating peptides interacting with the membrane, it causes outward curvature. You can get continual outward curvature as more and more of these peptides, the VMP-labeled fusion proteins, interact with the membrane at that location. You'll then get the formation of the vesicle. Once that you got inward curvature of the membrane of the vesicle itself, that's when you'll get release of the protein. So it all stays within the lumen, and eventually, once at a critical point in terms of the size of the vesicle, once you reach a critical size, that vesicle gets released into the media, and see, off it goes. Okay. And we've got some really nice images I think about to show you now. Okay, so here's just another image. So again, this is a super resolution image where this time we've got the vesicle nucleating peptide is labeled with a red fluorescent protein. So labeled pink here is mCherry2, nice and bright. And here we've got the inner membrane has been labeled with the M neon green. So this is a sim image to show that the E. coli cells, and it's producing these vesicles, which are suddenly all that the recombinant protein suddenly gets exported into it. And we can see that in the next image here. Just see it form now, and you can see the protein all go into it. You can just see it there. I'll run that movie again. I'll pause the movie there, so you can just see the vesicle just starting to form. And then it exports out, and the protein all gets exported into it. It's like it exhausts into the vesicle. So there we go. And yeah, we've got ongoing things. We're trying to understand how further. So we've identified lots and lots of vesicle nucleating peptides through mutant screens and site-directed mutagenesis. to identify new peptides, and that's identified residues that are really important for enhancing the vesicle production. And there are residues that inhibit the vesicle production or inhibit the export. So they're really interesting as well and really important to us. Some of those vesicles nucleating peptides, we've identified they're really good at a variety of temperatures, whereas our initial ones they only really worked at 37 degrees. Now we've identified peptide sequences that work down to 25 degrees and even lower now. But these are all described in the recently published paper. So, and also some of these new ones. So we've compared the yields of some of our therapeutic proteins we're interested in, and Fujifilm are interested in, and we see a huge increase in yield. So here's, for example, let's look on the histogram on the left-hand side. On the left-hand bar, we've got DARPin by itself, 50 milligrams per liter of shaking flask culture. Remember, these are shaking flask cultures in the lab. Here's our initial construct of initial VMP, getting about a gram per liter. Then with our new iterations, we're getting 2.2 grams per liter. Oh, so overall, almost 2.5 grams of protein. Similarly, with Stefin, we're getting something like 2.8 grams per liter. And now we're finding that with some of the latest iterations of the peptide, we're getting well over 3 grams per liter. So these are experiments that we've undertaken, obviously in our lab. Other labs around the world, so with academic labs, so I've also tested this, but also Fujifilm. Obviously, they want to confirm this because they want to put this into their system, and or they're interested in it, and they've been able to reproduce these results, almost getting identical numbers. So it works in both an academic and a commercial environment. So finally, we've identified this vesicle packaged recombinant protein system. It's required for both. It's important for both production of the protein because it increases the yields, but also that export into the vesicles simplifies the downstream processing. And also these vesicles, they can be used for diverse applications. We're routinely obtaining yields of over 3 grams per liter now from shaking flasks. So it's significantly enhancing the yield of functional protein, so these vesicles provide a vehicle for long-term storage. So what we do is we isolate them, we purify them, and leave them in the media, and we just put them in the fridge, or we exchange them in something like PBS, a buffer. Depends on the proteins inside. So we got really simplified downstream processing. The system allows expression of many proteins which are toxic to the bacterial cell. We can produce proteins that have disulfide bonds, like antibodies, that we've been able to produce, and isolate. Allows isolation of vesicularized complexes, like inside the cell, like a number of antibodies that we're actively going. I haven't really had a chance to talk about those today, but they were included. Yeah, they're included in the patent, so you can look it up there. And the most important slides of all, the people who did all of the work. So this work was initiated about 7 years ago by a master's student, Tara Eastwood, when she came into the lab. So she's absolute joy to have her. And she's continued in that. She undertook a master's, a PhD, and a postdoc, and now she's undertaking her second postdoc. Second postdoc in the lab. So Karen she's been in the lab for a long time as well, and she's been involved in the later stages of the project. We've our close collaborators at Fujifilm Diosynth. There's Chris Lennon, you can see in the image here. So this is where Karen went up to Fujifilm to go and learn how to use the AMBER system up there. But also some of the images I showed you, so the other collaborators, really important in this collaboration, has been the ability to do some really high-end super resolution imaging, and that FLIM FRET experiment I was undertaken. So that was undertaken at the STFC funded central laser facility at Harwell here in the UK. And that was with the help of Lin Wang on the SIM images and Professor Stan Botchway for the images for the FLIM FRET experiments as well. So with that, thank you very much. And I'm happy to take any questions you may have.
All right, thank you, Dr. Mulvihill, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Let's get started. All right, our first question is this technology free to use?
Okay, so it's free to use to academics. Obviously, it's a part of our research funding. We need to be able to make it freely available to non-profit organizations and academia. However, that we have got ongoing collaborations with a number of industrial partners. So we need to get an agreement in place and licensing agreements in place, and that's worked very well for us and for everybody in the past. And we've had some quite exciting and collaborative projects using this technology. So yeah, it's not free to use, but to commercial sides, but we can find a way through it. Normally we can.
All right, great. Next question here. Have you tried applying this technology to other cell systems?
Yes, we have. Excuse me. So we've tried it on other eukaryotic systems and other prokaryotic systems, and it works very well in a number of the prokaryotes we've used. We've tried it other gram negatives. It works really nicely, and some extent in bacillus, so gram positives. We've looked at yeast systems, and we do see some evidence of it working there as well. So we're working on Pischia, and we've looked Pischia pistoris and in Schizosaccharomyces pombe. We've looked at it there, and also we've looked at mammalian expression systems there. So it worked. We've got some really nice preliminary data that we're looking forward now. It's included in the patent, but so we're happy to explore and exchange, explore ideas with people. But so if you want to look up more details on it, but it works with hex cells as well.
All right, great. And we'll go ahead and wrap up with this final question. Does it work on all proteins?
Okay, so I mean, as I alluded to during the presentation, some proteins get exported, some don't get exported, so they get internalized. Okay, so it doesn't work. It doesn't get exported for every protein, but I can say for every protein we have examined to date, we get an increase in yield of those proteins. It's been dramatic, a significant increase over whatever anyone has been able to get before with those proteins. So it's worth trying. It takes some tweaking. It takes some trying with the different peptides and different growth conditions, but it does work for every protein. It enhances the expression of every protein we've looked at so far. Okay.
All right, great. Well, thank you again, Dr. Mulvihill. Do you have any final comments for our audience?
No, it's just, yeah. So if you want to use it, please, go and read the paper about it. It's out there. It's open access. The constructs are freely available on AdGene if you are an academic or a non-profit organization. If you are a commercial organization, just get in contact and we can see what we can do.
All right, great. Well, thank you again, Dr. Mulvihill, for your time today and for your important research. Before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions we did not have the time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provide at the time of registration. This webcast can be viewed on demand for 2 years until April 5th, 2025. LabRoots will alert you via email when it's available for replay. We do encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.